Hi there, my name is Hannah Gannon, and today I'm going to be discussing antibiotic, penicillin, and also cover the topic of antibiotic resistance and how this has developed and how it's a very, very pressing issue in the public health and medical community as well. To begin, my name is Hannah Gannon, and we're just going to jump into what exactly penicillin is. So penicillin is an antibiotic, and it was first discovered in the late 1920s, and it belongs to the family of, it's the beta-lactam family. And so this is a group of antibiotics, and they specifically function by inhibiting the synthesis of a bacterial cell wall, and as a result, the bacteria ends up dying because it's not able to synthesize that specific wall, which is extremely important for rigidity and overall structural support for that bacteria to survive. All right, so now I'm going to talk a little bit about how exactly penicillin works and the mechanism by which it works as well. So, one of the primary components of a bacterial cell wall is a macromolecule called peptidoglycan. And peptidoglycan is extremely important for the bacterial cell wall. It provides, rigidity and support to it. And one of the final steps of producing the cell wall is actually creating this cross-link between one peptidoglycan chain, and it's going to be cross-linked to another peptidoglycan chain. And it's going to do so through an enzyme, and this enzyme is called a transpeptidase. And in more general terms, it's referred to as penicillin binding protein. But in today's lecture, I'm going to probably refer to it mostly as transpeptidase. Now, in terms of how this mechanism specifically works, so generally, when you're producing the bacterial cell wall, the typical synthesis operates one of the last steps of this is an amino group at one end is going to actually attack the peptide bond between two D-alanine residues, and alanines are just amino acids. So it's going to attack the peptide bond between these residues and another peptide unit, and it's going to use this transpeptidase enzyme to do so. And what's really unique about how penicillin operates is that penicillin actually resembles the D-alanine residues through this structure of a beta-lactam ring. And so that's something that's structurally significant to the penicillin, is this beta-lactam ring. And so overall, penicillin contains this beta-lactam ring, and it's able to act as almost or resemble the substrate for transpeptidase enzyme, and it's going to operate by irreversibly inhibiting the enzyme and therefore stopping the synthesis of the bacterial cell wall and cell growth. And as you can see, this mechanism just kind of emphasizes that. So here is the active site of the transpeptidase, here's penicillin, and it's going to attack that beta-lactam ring, and then you're going to block the synthesis of the bacterial cell wall, and as a result the bacteria will end up dying because it's going to be vulnerable to outside pressure and water, and so it's going to die pretty quickly. And one thing that I did want to mention that's really important when discussing this topic is that there's several different types of bacteria. There's what's called gram-positive bacteria and gram-negative bacteria. And gram-positive bacteria contain a higher amount of peptidoglycan. And as a result, you're going to have a higher concentration of this transpeptidase enzyme as well. And so that's why penicillin is more effective against gram-positive bacteria compared to gram-negative bacteria. Gram-negative bacteria have a thinner cell wall because they contain less of this peptidoglycan macromolecule. And all right, next I'm just going to jump to talking about a really pressing issue within the medical community and the public health realm, and that is antibiotic resistance. So In 2014, the World Health Organization actually said that antibiotic resistance is becoming a worldwide threat to public health, and this is due to a number of reasons. It can be due to, like people not finishing their full course of antibiotics and these bacteria just developing different mechanisms of resistance over time, and also like overprescribing these antibiotics. And so that's something that's definitely being more monitored, I think, nowadays, is just really making sure that you're not overprescribing antibiotics, because they are able to develop resistance fairly quickly. And so, like I said before, there are a number of ways that bacteria can develop resistance to antibiotics. Now, one specific mechanism that I did want to touch on today was how some bacteria create a bacterial enzyme that's called a beta-lactamase, or in some cases it's called a penicillinase, and these act to destroy the beta-lactam ring of penicillin, and therefore it makes it ineffective in killing the bacteria. And the specific mechanism by how beta-lactamase works is by hydrolyzing the peptide bond in the beta-lactam ring of penicillin. Now, something that scientists have worked to do to kind of combat this is create artificial or synthetic penicillins, and so that's something that's currently being in use today. And one last thing that I did want to touch on as well is one type or specific type of bacteria, which is called MRSA, and it is actually known. It's really serious, because it's actually known at being resistant to all of the different beta-lactam antibiotics. And so this is something that is definitely concerning, because usually if what bacteria is antibiotic, or if it's resistant to like one type of antibiotic, usually you can like use a couple or like a cocktail mixture, but this is something that is definitely concerning, because it's forcing us to create other alternative treatments for bacterial infections specifically. All right, so to finish up, I did want to just show kind of a model of what penicillin looks like in the active site of the penicillin binding protein, or transpeptidase, and kind of how this, and then just kind of reiterate how this works to inhibit the synthesis of the bacterial cell wall. So, okay, so as you can see here, we have penicillin, and it doesn't have the beta-lactam ring. It's actually not fully, so if it did have the beta-lactam, it would be formed right here, as you can see in the structure. And so this is in the active site of the enzyme, and then if I zoom out, you can just see more of that protein there. So you can see more of like that 3D structure with the different secondary, protein structures, like the beta pleated sheets and the alpha helices and whatnot. So that's kind of how it would look, which is pretty fascinating that we have the technology to create such detailed images. And so, to reiterate, I just wanted to say that the penicillin, again, is going to bind to the active site of the enzyme, and it's going to resemble the substrate, the typical substrate of this transpeptidase enzyme, and it's going to inhibit it. It's going to irreversibly bind to the enzyme, and it's going to therefore prevent bacterial cell wall growth, and as a result, the bacteria will end up dying just as a result of losing that, structure and rigidity that the bacterial cell wall typically offers. And let's get back to present. Let's see. Okay, and then just to finish off, I just wanted to show my references and where I got like all my pictures for this presentation today. So, in summary, penicillin is one of the earliest forms of an antibiotic, and it operates by resembling the substrate of a transpeptidase enzyme, which is used in the synthesis of a bacterial cell wall, and it's going to irreversibly bind to that enzyme, and it's going to as a result inhibit the synthesis of the bacterial cell wall and lead to that bacteria end up dying. And something that's been a pressing issue as a result of all these antibiotics being used today is antibiotic resistance, and so that's just something that's going to be interesting to see in the near future how we combat that, because it is a big major public health concern today. So thank you for listening to my presentation.